Hello, this is Gina Piscatelli with a presentation for an ear lab. In this lab, we will learn the anatomy and physiology of the ear. You'll learn three major regions of the ear, the structures inside each region, and how sound is detected and transmitted to the brain. Now, the ear is also involved with detecting the position of our head, detecting gravity, and sensing our equilibrium in the environment, our balance in the environment. And we'll discuss what regions perform these functions, but we won't learn the mechanism precisely. So please use the learning outcomes listed in your lab manual as a study guide to prepare you for the quiz that you're going to take after this lesson. Notice that the first two learning outcomes focus on anatomy of different regions of the ear. Number 1 says identify structures on diagrams of the whole ear, and then number 2 specifically asks you to pay attention to structures in the inner ear, including a cross-section through the cochlea. And then learning outcomes 3, 4, and 5 deal with the functions of the ear. We'll learn that there's a region called the vestibule, the cochlea, and then another region called the semicircular canals, and each of those perform a different function. And then number 4, notice that it says we're going to look at the function of certain structures, very specific structures, during hearing. And then number 5, we'll talk about hearing loss a little bit. So to begin, let's contrast the ear with what we've already learned in the eye. Like the eye, the ear is a special sensory organ. So in the eye, the cells that act as sensory receptors are called photoreceptors, specifically rods and cones. These photoreceptors are stimulated by light waves. In the ear, the cells that act as sensory receptors for sound and equilibrium are called hair cells. So there's a picture of a hair cell on the right of this slide. And notice on the diagram that the hair cell has structures called stereocilia. They look like little hairs. That's why the cell is named a hair cell. However, these stereocilia are not proper cilia that we learned about before, which are made up of microtubules. Instead, these stereocilia, although they appear similar, are actually extensions of the cell membrane, so there aren't microtubules in there. Also notice that the hair cell is in close contact with a neuron, labeled on the bottom of the diagram. When the hair cell becomes activated by a sound, it releases a neurotransmitter, and that neurotransmitter excites the sensory neuron. It's the sensory neuron that will transmit a signal to our brain so that we can perceive and recognize sound. There are similar hair cells for detecting balance, equilibrium, head position, and they function very similarly and will also be connected to a neuron that transmits a signal to our brain. Okay, we're going to begin our study of the ear by looking at the anatomy of the entire ear. There's three main regions. On this diagram, notice in pink, or the pink background, that indicates all structures that are part of the first region called the external ear. And then blue indicates the region of the middle ear, and green indicates the region of the inner ear. So the external ear includes the pinna or the auricle, the external auditory meatus, Sometimes we just call that commonly the ear canal, and up to the tympanic membrane, which we commonly refer to as our eardrum. So this region of the external ear is filled with air. Now let's look at the middle ear. The middle ear extends from the tympanic membrane to the vestibule. And the vestibule is this part of the inner ear, but there's a wall there. So you can kind of see that this is the wall of the vestibule. This part is entirely, this middle ear, is entirely embedded in the temporal bone, a skull bone. And it includes the auditory ossicles, which we learned about, and an opening that goes into our pharynx, high up in the back of our throat. And this is called the eustachian tube right here. Now, the inner ear includes three main structures, and these are all also embedded in the temporal bone. The inner ear includes the semicircular canals. There's three of those, orientated in different positions, lateral kind of, and then superior, inferior, and then there's one for kind of detecting anterior, posterior, but we won't get into too much detail with that. It also, in addition to the semicircular canals, the inner ear has the vestibule and the cochlea. Notice that both the vestibule and the cochlea have connections with nerve fibers. So the nerve that leads from the vestibule would be called the vestibular nerve, and the nerve that leads from the cochlea would be called the cochlear nerve. They eventually merge together to form cranial nerve number 8, which is called the vestibulocochlear nerve. And we'll talk about that again in a little bit. Okay, let's just focus on the outer and the middle ear to begin with. And as I talk about the structures, I'm going to talk about their function a little bit as we go. So the auricle, or the pinna, whichever you wish to call it, helps direct sound waves to the external auditory meatus. And that red wiggly line that's already drawn on the slide is supposed to represent a sound wave. So the sound wave goes into the external auditory meatus and causes the tympanic membrane to vibrate. So it really does act like a drum. It kind of moves in and out, in and out, in and out with the sound wave. Now that movement of the tympanic membrane causes the auditory ossicles to vibrate. And the three auditory ossicles that we learned in the skeletal system are the malleus, the incus, and the stapes. The malleus is physically attached to the back of the eardrum. And there's a joint between the incus and the malleus, and between the stapes and the incus. So as the tympanic membrane vibrates in and out, the malleus hammers back and forth against the incus, and that causes the incus to vibrate, and it kind of acts like a lever and push the stapes towards the vestibule and then away from the vestibule. And we'll come back to how that works in a little bit. So for the outer ear, we have the auricle and the pinna and the external auditory meatus up to the tympanic membrane. And then in the middle ear, we've talked about the tympanic membrane and the auditory ossicles. There's one other structure that I'd like to point out in the middle ear called the eustachian tube. The eustachian tube is like a canal between the middle ear and our pharynx. And what it does is it equalizes pressure on both sides of the tympanic membrane. If the middle ear was closed off, then it wouldn't have access to atmospheric pressure. It would have to have its own pressure filled with air, and that would likely not change with atmospheric pressure. And as you know, we um, we change our altitude as we go up hills or down hills or in an airplane, and that changes the atmospheric pressure in the external auditory meatus. And to make the tympanic membrane function properly, we need pressure to be equal in the external auditory meatus and the middle ear. So the eustachian tube, by opening our mouth, of course, and because we have nostrils that lead to the pharynx, by having this eustachian tube linking our pharynx to the middle ear, now the middle ear has contact with atmospheric pressure, and those two regions will have equal pressure in the external auditory meatus and the middle ear. Okay, now we're going to look at the middle ear and its connection to the inner ear. Once the sound wave has caused the auditory ossicles to vibrate, what happens is the stapes pushes against a membrane that's in the vestibule. Now we can't see this membrane on this picture. I'll show you another picture later because the stapes is covering it up. But it's like a window with a thin film over it, and it's called the oval window. So the stapes will push into the oval window and then pull back and release it, and then push in, pull back, push in, pull back with the sound wave. As that happens, the oval window, which is a membrane, presses into the vestibule, which is part of the inner ear. Now, the inner ear is completely closed off from the atmosphere, so there's only fluid in the inner ear, not air. So as the stapes presses against the oval window, the fluid in the inner ear will move. Okay, let's look at the structure in more detail of the inner ear. We have 3 semicircular canals. Those are responsible for sensing the position of our head relative to gravity. And then we have the cochlea, which is responsible for detecting sound. The vestibule is kind of involved with both. So it has functions in hearing and in equilibrium. But most importantly, it has that oval window with a membrane that connects the inner ear to the middle ear, and the stapes presses against that oval window. I want to point out also that there's another membrane that helps kind of relieve the inward pressure of fluid. It's called the round window. So when fluid is pressed, or when the stapes presses against the oval window and the fluid begins to move, it will eventually cause this round window to bulge outward. One thing that's important in getting the fluid to move in the inner ear is to make sure there's enough force generated so that the stapes has enough force behind it to push into that oval window and cause the slightly viscous fluid of the inner ear to actually begin moving. Now, if a sound wave traveled directly to that oval window and we didn't have this tympanic membrane and we didn't have the auditory ossicles, that oval window's surface area would be so small that I don't know if it would collect enough force to cause the inner ear fluid to move. So instead what we have is a larger surface area membrane called the tympanic membrane that captures the force of sound waves and causes these auditory ossicles to transmit that great quantity of force to a very small surface area. So one of the functions of the auditory ossicles is not just to cause the oval window to be pressed, not just to press against the oval window, but also to transmit the amount of force that the tympanic membrane experienced. So this is a different diagram of the inner ear, and it's almost a cross section so that you can see inside a little bit. And you'll notice that although the inner ear is housed in bone, there's also some soft membranes present, and those are indicated in blue, and they're fluid filled. So the inner ear, as I mentioned before, is made up of the semicircular canals, and they have special hair cells and structures that detect rotational movements of our head in different planes. When our head moves in different planes, different semicircular canals are stimulated. All by fluid movement, this neuron will be stimulated and it'll be transmitted to the brain. So it's still fluid filled, and it's going to work very similarly to the way the cochlea works for hearing. Okay, so the cochlea is responsible for detecting sound waves, and those sound waves get transduced into fluid waves that excites hair cells, and the hair cells send a signal along the nerve. And here's where the two nerves, the vestibular nerve and the cochlear nerve, merge, traveling within the temporal bone still, but eventually to the brain. And that nerve is called cranial nerve 8 and named the vestibulocochlear nerve. Now, the vestibule is kind of interesting because the vestibule also has specific hair cells. Not only is it a location where the stapes, presses on this fluid that's right here, but there are sensory hair cells in the vestibule proper, and they're labeled as purple here. They have specific names, but we're not going to cover them. When the fluid moves due to changes in head position as well as gravity, they are excited. So they're kind of part of the semicircular canal function of equilibrium, and signals will be transmitted via the vestibular nerve to cranial nerve 8. Because we're going to focus primarily on hearing as the function of the inner ear, we need to look in detail at the anatomy of the cochlea. So the cochlea looks spiral, of course, and it is coiled up. If it were straightened out, it would be about 34 millimeters long. And within the cochlea are tubes filled with fluid. And the tube that contains the sensory hair cells is called the cochlear duct. And it's right in the middle here. So the cochlea uncoiled resembles a tube within a tube. So the outermost or peripheral tube in this picture is purple. It's uncoiled, of course. But inside is another tube, which I've drawn as blue. And it is inside, but I've made it so that you can actually see it, kind of like cut through the peripheral tube so that you can see into the inner tube. All regions are filled with fluid. Okay, and because there's an inner tube inside of an outer tube, there's different regions that are formed. The region on top of the cochlear duct or the inner tube is called the vestibular canal. This is continuous with the membrane of the oval window that the stapes presses against. And then we have the inner tube, which is called the cochlear duct. And then notice that the vestibular canal goes kind of farther than the cochlear duct does. It is continuous with the space underneath the cochlear duct, and that's called the tympanic canal. Now, since all three regions, the vestibular canal, the cochlear duct, and the tympanic canal, are filled with fluid, fluid movement is going to happen first in the vestibular canal. And in some cases, that fluid movement is going to happen all the way to the apex here of the cochlea and transmit within the tympanic canal. The tympanic canal is continuous with the round window, and so when fluid in the tympanic canal moves, this round window will bulge into the middle ear. It'll bulge to the left. Now, not all fluid is the same in these three different spaces. The vestibular canal and the tympanic canal are filled with a fluid called perilymph, peri meaning outer, and it has a certain ion concentration that differs from the fluid that's in the middle space called the cochlear duct. That fluid is called endolymph. And again, it has a different ion concentration, but we aren't going to worry about those different ions. Now, the cochlear duct is closed on both ends. Okay, it's not open to another space. So the only way that the fluid will move in the cochlear duct is if there's a downward pressure or an upward pressure of fluid bulging into the cochlear duct. And this is really important in how hearing, the function of hearing occurs, because within the cochlear duct is a membrane, and it's kind of light purple or pink that I've underlined here in red. That's called the basilar membrane, and we're going to look at that in more detail. This is where the hair cells sit. They sit right on the basilar membrane. And so anytime there's a movement downward into the cochlear duct of fluid, pushing on the cochlear duct, or upward, these hair cells are going to move. And we'll talk about how they become excited, which movement causes excitation of the hair cells there. And we're going to do that next. So to understand how the hair cells sitting on the basilar membrane of the cochlear duct are excited, we really need to look at not an unrolled version of the cochlea, but we need to look at how we need to look at a cross section of it. We need to look at one region right here, and how these spaces are partitioned. How is the vestibular canal separated from the cochlear duct, and how is the cochlear duct separated from the tympanic canal? Well, as you might have guessed, there are epithelial membranes between each region. And so we're going to look at what's the name of this membrane between the vestibular canal and the cochlear duct, and the name of the membrane between the tympanic canal and the cochlear duct. So this is an enhanced view of a cross-section through the cochlea. And let's just do anatomy first. We have the vestibule or vestibular canal with paralymph on top here in this diagram. The tympanic canal, the lowest region with paralymph that we've already talked about. And then we know the cochlear duct is in the middle, and the cochlear duct contains endolymph. That's what we know so far. Now the new stuff. The membrane between the vestibular canal and the cochlear duct is called the vestibular membrane, right here. The membrane between the tympanic canal and the cochlear duct we refer to as the basilar membrane. Now it's this basilar membrane that has some cells that are connected to neurons. Those cells we've already learned are hair cells, but the hair cells are not individually placed on the basilar membrane. Instead, the hair cells are grouped together in a unique structure called the organ of Corti that's circled in this diagram. In some textbooks, the organ of Corti is called the spiral organ. So just note the positions of where the vestibular membrane is, that the organ of Corti lies in the cochlear duct, and that the basilar membrane is what the organ of Corti sits on. Now, the organ of Corti contains the hair cells. Those are the sensory cells. So we know that the organ of Corti and the hair cells, when stimulated, are going to send an impulse to the cochlear nerve. Okay, to understand how the hair cells and the organ of Corti function, how they become excited to even send a signal into that neuron of the cochlear nerve, we need to look at the structure of the organ of Corti in more detail. So this is the vestibular membrane that I've just drawn an arrow to, okay? And we have the basilar membrane sitting underneath the whole organ of Corti. Now, the organ of Corti is made up of rows of hair cells. There's three rows of hair cells. We call those the outer hair cells. And then there's one that's more medial, and we call that the inner hair cell. There's also some supporting cells in this organ of Corti that's sitting on top of the basilar membrane. Notice that the hair cells have that stereocilia that we identified already. And then there's one other structure that's interesting, and it's called the tectorial membrane. The tectorial membrane doesn't sit on the basilar membrane, but it is considered part of the organ of Corti. Instead, it's attached to the medial part of the cochlea, and it's quite stiff. Okay, so it's kind of like a shelf reaching into the cochlear duct. So the hair cells are the sensory cells for hearing, and they become excited when they press up against the stiff tectorial membrane. And that's because the stereocilia bend. So the hair cell moves up towards the tectorial membrane, the stereocilia bend, and that excites the hair cell, which then releases a neurotransmitter that excites the neuron. So we're going to look now at the big picture of hearing, how the sound wave that comes through the external auditory meatus actually results in excitation of the cochlear nerve. So here's the big picture on the physiology or function of hearing, how hearing works. Sound waves that are traveling in air, so waves in the air with certain frequency and amplitude. Amplitude is like how high are they, and frequency is, how often do they occur? Are the waves happening tightly together or more spread apart? But nonetheless, these sound waves traveling in air travel through the external auditory meatus. And because of the function of the middle ear, those sound waves are transduced or turned into fluid waves in the cochlea. Now, the movement of fluid in the cochlea will activate hair cells, and those hair cells will activate neurons, but the neurons still have to travel to the brain so that we perceive the sound. And the part of the brain that recognizes sound that the cranial nerve 8 travels to is the temporal lobe. So hearing, or the auditory cortex, is located in the temporal lobe of the cerebrum. So step by step, we're going to go through how sound waves are transduced into fluid waves. And this diagram you have in your textbook. It's in chapter 15 on page 587. But you have another one in your lab manual that you can use as well. So the first thing I want to mention is that when sound waves hit the tympanic membrane, not all sound waves are perceivable for us. Like we can't hear really high frequency sounds like dogs can, for example. And so what happens in that case for sound waves that we really don't perceive or detect really is that the sound waves come in and they vibrate the tympanic membrane. The auditory ossicles will still vibrate, and there is pressure waves in the fluid of the vestibular membrane, of the vestibular canal, all the way out to the apex, in the tympanic canal, and the round window will bulge towards the middle ear. But the cochlear duct is never affected. So sounds that are out of our hearing range travel through this apex, which has a fancy name called the helicotrema, and these fluid waves never press into the cochlear duct. But for sound waves that come in that we can perceive, what happens instead is that these sounds within hearing range travel again in the vestibular canal, but they press on the cochlear duct. That causes downward displacement of the basilar membrane. And eventually we're going to get excitation of the inner hair cells. So, I better erase this. Hang on. So we're going to go through physiology of hearing, something that's in our range of hearing instead. So the first step is the stapes presses against the oval window. The oval window then bulges into the vestibular canal. That's labeled as number 2. This causes the perilymph in the vestibular canal to flow or have a fluid wave. The perilymph in that vestibular canal then presses downward on the vestibular membrane. That's labeled as number 4. Then the endolymph in the cochlear duct moves. That's number 5. And causes the basilar membrane to be displaced downwards into the tympanic canal, and that's 6. Now you know the hair cells are sitting on this pink line. That's where the organ of Corti is, but they have not been excited yet. In fact, the hair cells have been moved farther away from that tectorial membrane of the organ of Corti. So let's look at the next thing that happens, because it's the recoil of the fluid movement that's going to excite the hair cells. So when the basilar membrane is displaced downward into the tympanic canal, now the perilymph in the tympanic canal experiences a wave of pressure, and it flows towards the round window. The round window then bulges into the middle ear because it's just a epithelial membrane. Then the next thing that happens is recoil. So the recoil happens in part just because this round window will recoil back into the tympanic canal, but also because the stapes doesn't press continuously forward into the vestibular canal. Instead, it also pulls backwards. So when the stapes pulls backwards, that helps the recoil of the round window. And so the round window bulges into the tympanic canal, and the perilymph begins to flow backwards. So we have number 9, where the stapes moves back from the oval window. That helps with recoil of number 10, the round window bulging into the tympanic canal. And then number 11, we have the perilymph flowing backwards or to the right into the tympanic canal. What this causes is upward displacement of the basilar membrane. And that's going to make the organ of Corti, which is sitting on it, and the hair cells move up. And the hair cells will then press on the tectorial membrane. So we need to look at a different picture so we can see the hair cells. So at rest, this basilar membrane is kind of flat. The hair cells are touching the tectorial membrane, but the stereocilia aren't bent. But as soon as that perilymph from the tympanic canal experiences a wave, it presses up against the basilar membrane, and the basilar membrane is displaced upwards. This causes the whole organ of Corti, except for the tectorial membrane, and the hair cells in particular, to move upward as well. And so the hair cells press on the tectorial membrane, and the stereocilia they have bend. So the stereocilia bending is what is going to excite the hair cells. And the way that happens has to do with an ion channel. So when the stereocilia of a hair cell, shown here, a hair cell shown here, when the stereocilia bend, these potassium channels open up and potassium floods into the hair cell. That causes the hair cell to release neurotransmitter to the cochlear nerve or neuron. That cochlear neuron then is excited by the neurotransmitter, and it will eventually send a signal to the brain. So you do have an exercise in your lab manual asking you to retrace this pathway using a diagram in your lab manual. It's like this, and you're going to study the sequence of events involved in how sound waves stimulate hair cells and the basilar membrane. And so you'll just trace the stapes movement, the oval window movement, wave in the vestibular canal, etc., so on and so forth, and then the recoil and upward displacement. And I'll have an answer key available for you as well, which is essentially also on these slides. Okay, the last thing that we have to do for this lab deals with your fifth learning outcome on the study guide, dealing with types of hearing loss. And there's two types: conductive hearing loss and sensory hearing loss. Conductive hearing loss occurs when we don't hear because a sound to the inner never reaches the inner ear. Transmission to the inner ear is somehow blocked or compromised. So this can happen if the tympanic membrane has a hole in it, it's perforated, and so it doesn't vibrate correctly. It could also happen just due to plugging the external auditory meatus, so the sound wave can't even reach the tympanic membrane, like earwax, but that would be an awful lot of earwax. Okay. More commonly, conductive hearing loss happens due to malfunction of the auditory ossicles. When there's an middle ear infection called otitis media, you get inflammation in the middle ear, and when you get inflammation, there's fluid in there, and so the auditory ossicles aren't going to move as well. You'll probably still hear, but it'll be muffled. Alternatively, if you have chronic inflammation, you can develop an arthritic condition where the auditory ossicles, as well as the cochlea, develop these bony outgrowths. They're shown here in like white. Okay. And what happens is this bone fuses the stapes to that oval window or vestibule, and so the stapes just can't move anymore. Now, this is treatable, and I should have said that when the auditory ossicles are fused like that, it's called otosclerosis, but this is treatable. Otosclerosis can be repaired usually by just surgically implanting a stapes, because it's usually the stapes that's the problem. But it's an artificial stapes made out of titanium usually. So that will help with, and then of course you can clear out earwax and you can repair a tympanic membrane as well. Okay, the last type, or the second type of hearing loss, is called sensory hearing loss. And sensory hearing loss is the category that we would put any kind of deafness into that deals with the nervous system. So neural transmission is compromised. That means from the hair cells to the cerebrum. So when we lose hair cells due to the aging process, because some hair cells just die naturally, When old people can't hear as well, and I'm not there yet, but it won't be long, that's categorized as sensory hearing loss. We can also lose stereocilia, or the hair cells will be damaged at least. Maybe they don't die, but the stereocilia are damaged on the hair cells due to really loud sounds. Like, don't listen to the radio too loud, or a bomb exploding, or something like that. That would be categorized as sensory hearing loss because the hair cells won't function properly. Of course, anything that causes damage to the temporal lobe of the cerebrum, like a traumatic brain injury, a concussion, something like that, maybe a surgical intervention for epilepsy, anything like that could also cause sensory hearing loss. And then there's also the case of just plain old congenital defects. And in that case, there's probably not proper development of the cochlea. Or I suppose it could also be proper development of something in the middle ear. But generally speaking, when someone is born with hearing loss due to improper development as an embryo and a fetus, often this can be treated by putting a cochlear implant in place. Now, cochlear implants are only going to work, generally speaking, if the nerve fibers are intact and the brain is intact. So you're going to have the nervous system proper, intact, and functional. So cranial nerve 8 has to work, and the temporal lobe has to work. But the implant detects sound waves and then stimulates the neurons or nerve fibers of cranial nerve 8. And there's different electrodes that are inserted in the round window. You don't have to know about that. And along the cochlea, spaced so that different frequencies can be detected. But the most important thing is to know, for you, is to know what is the difference between conductive hearing loss and sensory hearing loss. And conductive hearing loss, again, is anything up to the auditory ossicles. And sensory hearing loss deals with the cochlea, the hair cells, and the nerve and brain. Okay, the last thing that you need to do for this lab is complete the lab exercises in your lab manual, and then take a quiz on ear anatomy and ear physiology. If you have any questions, please attend the live lab session for your class. Thank you.